Welcome to This Explainer. Today, we're diving straight into a fascinating concept. What happens when we pull AI out of a mathematical void and drop it into a dynamic 3D physical volume? We're looking at cutting-edge research that completely reshapes how machines learn, not by just adding more standard neurons, but by building an entirely parallel biological system embedded right there in space, the glial network.
To really wrap our heads around this, we're going to build this biological ecosystem progressively, layer by layer. We'll start with escaping the spaceless void, move on to the astrocyte field, then introduce microglial agents, explore broadcast and volume transmission, and finally bring it all together in a new dimensional ecosystem.
All right, let's kick things off with Section 1, Escaping the Spaceless Void. This is all about moving AI from a null space into a physical volume.
So think about how standard artificial neural networks work. They exist in what we'd call a null space. When layer 1 computes a signal, it just instantaneously teleports over to layer 2. Right? There's zero concept of physical distance, and the inference is completely stateless. But real biology? It operates in a physical 3D tissue volume. Signals don't teleport, they physically travel, which takes a finite amount of time, and they interact with a continuously changing multi-timescale environment along the way. To fix this teleportation issue, researchers do something called a spatial coordinate assignment. Basically, imagine taking a rigid abstract graph of neural connections and literally forcing it into a simulated 3D physical geometry. Suddenly, every single mathematical weight in your network has a physical x, y, and z coordinate. Spatially close weights now have correlated gradients. We're literally taking a spaceless math equation and giving it a structured physical body. And this geometry is absolutely crucial because it's the prerequisite for everything else we're about to talk about. Moving right along to Section 2, the astrocyte field, where we flood the graph with fluid dynamics. Once we have that 3D space, we flood it with a continuous volumetric fluid that represents astrocytes. In a real human brain, a single astrocyte can contact up to 2 million synapses. It has this massive spatial domain of influence. So, in our emulated system, these astrocyte domains operate as a continuous 3D diffusion field. Picture these distinct mosaic-like territories of modulating fluids just wrapping around our neural network, slowly reacting to local activity over time. Now, there is a stark contrast when you compare this to how we normally optimize AI. Standard optimizers like Atom or RMS prop, they just tweak learning rates globally based on math and time. But these astrocyte domains, they provide spatially correlated modulation. If one specific region of our 3D network gets highly active, the local astrocyte domain reacts using simulated calcium dynamics. This creates a multi-timescale memory that hangs around long after the neural activity stops. They aren't just running a math optimization, they are physically gating whether a region is structurally allowed to learn at all. In actual biology, this gating is vital. Astrocytes release a chemical called D-serine, and without it, certain synapses simply cannot learn, period. The astrocyte literally gates Hebbian learning, so neurons can't wire together unless the astrocyte gives the green light, shifting us from a standard 2-factor learning rule to a biological 3-factor rule. Plus, this fluid environment mediates heterosynaptic plasticity. That means if one synapse strengthens, the astrocyte can actually weaken a totally unconnected but physically neighboring synapse just to keep things balanced. It's a lateral interaction based entirely on spatial proximity, which standard backpropagation literally cannot do. That brings us to Section 3, microglial agents, the network's mobile architects. Building on that astrocyte fluid, we now introduce our second layer of spatial complexity, mobile microglial agents. These are little agents that physically patrol our simulated 3-dimensional space, constantly surveying the territory. When a region of the network starts throwing high errors or conflicting gradients, these agents sense it and physically migrate toward the problem zone. It's a process akin to biological chemotaxis, kind of like a shark sensing a drop of blood in the water. But once they get there, they don't just impulsively delete things. They hang out, accumulate evidence over time, and then decide whether to prune conflicting connections or signal for new growth. This is a massive departure from how we currently shrink AI models. Standard magnitude pruning just uses a global threshold, blindly deleting weights that fall below some arbitrary number. Microglial pruning, on the other hand, is spatially targeted. It acts like a continuous neural architecture search. These mobile agents clear out dead, confusing connections and actively trigger signals for healthy regrowth. It's basically a self-repairing, self-diagnosing maintenance crew living directly inside the physical geometry of the AI. Next up, Section 4. Broadcasts and volume transmission. how we actually replace the backward path. This right here is the absolute biggest hurdle in AI optimization today. In standard AI, we rely heavily on backpropagation. We calculate an error at the end, and then we instantly teleport that exact math backwards through the entire graph to update the weights. But biological brains absolutely do not have a backward pass. A local synapse deep in your brain doesn't magically know the global error of your overall action. So, how exactly do local parts of a network learn efficiently without backpropagation?
Well, the biological solution, when mapped to our 3D model, is spatial volume transmission. Instead of teleporting an exact mathematical error backward, error signals diffuse as physical wavefronts through the spatial embedding. Imagine dropping a pebble in a pond. The error ripples outward as continuous concentric rings. It propagates right through the 3D space, interacts with those astrocyte spatial domains we talked about, and naturally attenuates over distance. It's a localized broadcast. This brilliantly bridges the gap between biological reality and local learning performance, entirely bypassing the need for a backward pass.
Finally, let's pull it all together in Section 5, a new dimensional ecosystem, bringing local rules to scale. When you combine all these elements, simply forcing the network into physical space naturally creates an elegant multi-timescale memory system. Neural activations fire in milliseconds. Then the astrocyte calcium state, acting as a sort of context window, updates over seconds. The actual network weights adopt over hours. And finally, the overarching glial network topology literally crystallizes over days. It's a gorgeous hierarchy of memory that just emerges naturally, simply because the system finally exists in space and time.
Now, you might be wondering, okay, simulating physical volumes, fluid dynamics, mobile agents, the compute for this must be absolutely insane, right? Well, here is a genuinely staggering fact. Research estimates that running this highly complex, self-repairing ecosystem could incur a maximum computational overhead of only about 20%. And because those microglial agents are constantly pruning redundant connections, this system might actually end up using less compute for better performance than our standard spaceless models. This could literally be the key to building hyper-efficient AI that runs right on our local edge devices.
Ultimately, this entire framework rests on one incredibly powerful prediction. It predicts that these spatial geometries, these glial mechanisms, aren't just arbitrary biological noise. They actually provide far greater computational benefit under local learning rules than they ever could under backpropagation. They're the specific missing ingredient that allows biological local learning to actually scale up and compete with artificial backprop. Which leaves us with this final provocative thought to take away from this explainer. If escaping that spaceless mathematical void and adopting physical, 3D spatial properties is the true key to unlocking the next generation of resilient AI, what other fundamental biological dimensions are we currently completely overlooking? Definitely something to think about as you watch the AI landscape evolve. Thanks so much for joining me, and keep questioning the framework.